So a lot of my research has focused on medulloblastoma. This is the most common malignant pediatric brain tumour. It usually occurs in children. The peak age of incidence is between the ages of 3 and 8. It occurs in the cerebellum, as David's already mentioned, at the back of the brain. But the big problem with medulloblastoma is at diagnosis, a third of patients are already metastatic. This means that the tumour has spread through the CSF. You can see the arrows and the flow of the CSF on this picture, or through the intraventricular system. We're still developing our understanding of that. But where it ends up is either forming laminar, so a sugar coating across the top of the brain or down the spine, or it forms solid mets. And these are called nodular metastasis. The problem with metastatic disease, which David's already alluded to, is it responds very poorly to current therapies. Patients with metastasis or tumour spread are given more aggressive treatment. So they're given high-intensity chemotherapy, and if they're over the age of 3, they're given even more radiotherapy. And the consequences of this are pretty devastating. It damages the surrounding brain, it affects their hormones and their IQ, and it causes peripheral nerve damage. So what we need are biologically relevant treatments. And through this, we hope that we will increase survival and also, importantly, increase the quality of life for survivors. So what do we know about the biology of the disease for medulloblastoma? So I'm not going to go into the types of molecular analyses that have been done, but a great deal of molecular analysis has been done. And the last 8 years, it's become much clearer. And medulloblastoma splits into at least 4 different subgroups. Within those, it becomes more complex, and there are other subtypes, but mainly there are these 4 clinical groups. And these are highly clinically relevant. I don't know which one I can point to. If you look at the top blue line, this is the Wnt subgroup. Under current therapy, these are essentially curable. However, you have sonic hedgehog in group 4 in the middle, and this is group 3. And the survival rates are very low for this molecular subgroup. But when we think about metastasis, actually it maps directly onto it. So what we see is in the Wnt subgroup, you don't tend to see metastasis. You will see it occasionally, but not very often. Sonic hedgehog, one of your intermediates, you see a low level of metastasis. The highest level is in this group 3, with the group 4 coming close behind it. So we know there are clear molecular differences indicating that the drivers of the tumour are different for each of these subgroups. Now, traditionally, if we're going to test drugs, what's been done is they've been tested initially in the lab in 2-dimensional culture. You take the patient's cells, you develop them so they'll grow as a cell line, and we grow them across the bottom of a flask in a solution that's physiologically matched to the tumour. The next stage of this is to take your cells into a mouse model. So you'll take the tumour cells and implant them, either as shown here in the brain of the mouse, and then if we label them, we can follow the cells as they migrate and metastasize down the spine. And this is one of my models in the lab. We can also do genetic manipulation. If we're really keen and we see a pathway, it's really important, we can make a genetically modified mouse. And so we alter it, and we can produce a colony of mice that will always have that defect that you see in the tumour, and you can use it for testing drugs. However, based on these types of studies, the success rate for new drugs in phase 1 clinical trials is still about 3%. And of the drugs that make it through the phase 1, only 1 in 10 of those drugs will progress to give patient benefit. And none of the drugs that are used to date were developed specifically for children. So they're not built on the biology of the tumour. So the aim of my research group is to develop new, more representative models And we're doing this by building 3D culture models. And what I mean by three-dimensional is that we grow balls of tumour cells. So instead of growing them flat across the bottom of the plate, we can grow a sphere of cells. So it's three-dimensional, it's much more representative of the tumour. We can also grow these on or within matrices, either a simple matrix, so where we're modelling things like the blood-brain barrier or basement membranes, but we can also tailor and build more complex matrices, and I'm going to tell you a bit about that. The important thing about these is that they're high throughput. We can either do them in 96 well plates or 384 well plates, as I've shown you here. This plate is the size of my hand. Each well that we grow our cells in are the size of your pinky. So in one plate, we can carry out experiments that equate to 384 mice. So we can get a huge amount of data in our representative models. It's far cheaper than using mice, and also the material that we put in is humanized. So we can model things that are much more relevant to humans. So the first that I'm going to tell you about a little bit more depth, this is being worked out by Sophie Roper, who's a PhD student in my group. And she's built some of these spheroid models. So what we do is we take our single cells, we allow them to form our spheroids just by stopping the cells from sitting down. The plate is a low attachment, so they can't sit on it. So they form a ball naturally. Sometimes we need to give them a few other factors to help this to work. And we've also realized that we need certain growth factors in there, but we've worked out that this works across all subgroups of medulloblastoma, although some of them form a little bit looser spheroids. We form our spheroids by day 4. By this point, the diameter of them is the maximum diameter to allow perfusion of drugs. So we can study drug penetration into these. We can also look at their growth, drug responses I've mentioned, and we can take them further and look at migration and invasion. When we look at drug response, if you look at these, we've got a grey line and then a black line. And the blue arrow that I've put on shows a distinct shift in drug response. So these tumours, when we grow them in 3 dimensions, are much more resistant to therapies. And if you look at the one on the right, you'll see actually we have a survival of a drug-resistant population. We don't see that in 2-dimensional culture. So we're starting to develop something that's much more clinically relevant. And we can image these as well. If you look at the one on the bottom right, you'll see that this ball of cells at the end is very resistant to chemotherapy. Whereas the ones on the left, where we see a response, they're falling apart in response to chemotherapy. So we can start to model this within our system. When we think about migration, we can take our cells, if I can get this to work. Is it going to work? Yes. Have you got it running on the other ones? Okay. So what we see here is the cells being placed on a basement membrane to mimic some of the barriers in the brain into the CSF or into the blood system. And you can see the cells start to migrate out and join together. So we can start to inhibit this process. What we also observed was that there were little tiny particles coming out ahead of the cells. So if you look more closely, I don't know how well you can see it on this, but there's some little dots there forming ahead of them. We were interested in what these were. So we think that these particles that are being released by the cells are these little blue membrane-bound particles that I'm showing you here. And these are called exosomes, and they're carrying material, and they're running ahead of the cancer cells, and they're signaling. And now exosomes are very common throughout the body. Most cell types will produce them, and they're found in all bodily fluids, which includes the CSF. And they have many important functions. But within cancer, we believe that these exosomes are actually transferring important messages. We think that they're going ahead, they're promoting tumour growth, they're enhancing migration and invasion, they're promoting therapy resistance, and they're also dampening down the immune response. So they're helping the tumour to grow. And this is an image showing some of the exosomes being released by our cells, these little green dots. So in medulloblastoma, we think that what happens is the tumour releases the exosomes, and these go ahead and prepare the metastatic site. And our proposal is to look at these. Initially, we're modelling them in cell culture, but we believe if we take them and we extract CSF from patients, we'll be able to isolate them and use that for early diagnosis and therapy selection. And also, we can use it for therapy monitoring. Alongside this, I've got another PhD student, Christine Matoko, who's a neurosurgeon, and she's looking to develop MRI imaging to detect these within the CSF, so we can use that prognostically and for therapy monitoring. The more advanced models that we're developing are our brain-inspired hydrogels. So here, what we're doing is modelling the microenvironment of the tumour. So not just a ball of tumour cells, we're allowing the tumours to develop in a matrix that is matched to the microenvironment of the brain where they grow. We're using the backbone of the brain initially, and then we're altering that to fit with the different components found in the different subgroups. And through this, we can look at cancer growth, we can look at movement, we can look at response to drug treatment, and also we can put immune cells into these models and build much more complex models. This should start right away. So this is one of our tumours growing within the matrix. And here you can actually see the full migration profile of the cells. So we can monitor that down the microscope, and we can also look at response of these down the microscope. So the final stage that I'm going to tell you about today is Masha's project, and Masha's here in the audience. And Masha has been looking at how the cells move from the tumour into the CSF. And she's been building a blood-brain barrier model so we can look at the migration of these purple cells through. And what we're going to do is combine that with our hydrogel mini brain models. And so then we can put together a brain tumour with the CSF, and we can model that migration. We can look at putting drugs into the system, and we can look at inhibiting it. So that's my kind of whistle-stop tour of what we do to model medulloblastoma. Just to put it in context, so I've told you kind of about, our work is very circular, and I've told you about the middle bit of it. We also do some work where we're developing prognostic markers, so we have some work that's candidate-based, where we have some hypotheses of things that we think might be involved in this process, and this is being undertaken by Alice, who's here in the audience as well, and Louisa. They've got some markers that we've shown to be prognostically relevant, and they are testing these within the model systems. We're also doing convection-enhanced delivery work, where one of the drugs that we tested in our system, which was highly effective, we've taken forward into preclinical work where we're looking at CED delivery. And we actually switched that to diffuse intrinsic pontine glioma because that was a better system. And we showed the same drug, which is a novel temozolomide analog, to be just as effective in DIPG, and we're working with that, and hopefully we'll bring it back to medulloblastoma. And finally, just to say thanks to everyone that's involved and our funders. Thank you.